Good evening, participants. To all of you. Hello.
Good evening, sir.
Hello.
Hello. Yes.
Good evening, sir.
Good evening. Am I audible to all of you?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Good evening, Dr. Pankaj Chetia.
Good evening, sir. Ashok.
Yes. All of you are ready?
Yes,
Yes,
sir.
sir.
Okay.
Yes, sir.
If some of you have attended the yesterday's session conducted by Dr. Pankaj Chetia from DDBJ University, you are already aware of the biomolecules, and also you know about a little bit of the concepts in molecular biology. It will be easier for you to follow today. So today we will be discussing on a topic that is one of the basic components of bioinformatics, that is biological databases and overview. So I would like to request all of you to switch off your audios so that there is no disturbances in between. And if you want to put a question, you can switch on your audio, then put up the question. And I will give you around 5 minutes time at the end of my session to ask questions. So for now, I'll be taking on one presentation, PowerPoint. Please listen carefully. I welcome once again all of you to this session. I'm happy that there have been many participants who were willing to participate in this program, but unfortunately we could not accommodate more than 100. And amidst us, I think we have participants from all across the state of Assam, and also few participants from other states of Northeast, and also a very few participants from Bangladesh. Welcome once again, all of you. Please wait a minute. The presentation is loading. So today's topic is biological databases and overview. Let us first discuss what is a database. A database simply is a collection of data where the data are kept in a structured form in such a way that it becomes searchable, and also the data are updated very frequently so that you can get access to the recent data. And also along with data, there are some cross-references, and in some cases there may be also citations and references for the data sources. So I will discuss the definition of databases. So as I said, biological databases carry information regarding the biomolecules and the related information as per the different kinds of biomolecules, their discovery, the related literature, related patterns, and many other aspects of biological value. Now, the creation of online databases in biology started towards the end of the last century when the omics technology started. As I refer to omics technology, you might have been already knowing about the different biomolecules and the related aspect of study of different kinds of biomolecules. So now we are living in an era of omics where biology can be studied at different levels. I'll come to that a few minutes later. So if you see the growth of the most important biological database, as is called as GenBank, hosted by National Center for Biotechnology Information, US, it grew since its establishment in 1994 in such a way that there is a logarithmic growth of the data resources available in the GenBank today. And today, every 15 months, the quantum of data in GenBank gets doubled. That is the kind of growth taking place in biological databases. Now, as I refer to the different omics technology, as I said, now biology can be studied at different levels according to our knowledge of molecular biology. And therefore, based on biomolecules, our study can be divided into different omics. Say, for example, if we study genes and genome, we can call the science of genes and genome as genomics. Similarly, there are branches called proteomics to deal with proteins and proteomes, transcriptomics to deal with transcript, that is messenger RNA, and the pool of transcripts, transcriptome. Similarly, we can also have separate branch called lipidomics, glycomics, lipidomics for lipids, glycomics for carbohydrates. Similarly, we can have metabolomics to study of the metabolic processes or the pathway of interaction between molecules. And finally, what we have been traditionally dealing with in biology, the functional biology, which is studied based on the empirical data that we observe externally. That can be named as genomics today. So as we see, there are different strata of classification in molecular biology, which needs the help of bioinformatics to study. Because to deal with the data, enormous quantum of data generated by these omics experiments, there's a necessity of use of computer to store, disseminate, analyze, and interpret such data. And there is the requirement of the creation and dissemination of resources in the form of biological databases. And that is the role played by biological databases in our understanding in omics technology. Now, why do we search the biological databases? The biological databases are searched for different. Let us resume it. So we need to go into the databases, basically search for information regarding the sequences that we have. Suppose in your laboratory, you have already obtained a new sequence out of your experiment. It may be a sequence, it may be a messenger RNA sequence, or it may be DNA sequence. It may be. Now, if you want to know whether this information or the sequence that you have is entirely new, or maybe you are having a sequence which is already known. So in that case, search with the databases to find out, is it already in the bank, or is it homologous or similar to some sequences already in the database? Or you can find out details about a gene. What is the nature of the gene? What is its functional importance? What kind of protein it codes for? And what are the publications related to the gene or the protein product? All these things can be known if we search in the biological databases for query sequence. I once again request the participants to switch off their audio. Now, most of the sequences and other information in the biological databases undergo a processing. The processing is called annotation. So annotation means simply you can say, you can compare it with labeling. Just as you label a diagram and the different components or the parts of the diagram is labeled properly by naming each of the parts and components. Similarly, the sequence may be annotated by identifying the different parts and also associating the structure as well as the different components of the sequences, and also associating the related publications which are already published in relation to the sequence. So this process is called annotation. Now, you can also search biological databases to find out the sequences which are similar to your query sequence. It will also give you an idea about the source of your sequence, or you can also identify the sequence. Similarly, you can search for some non-coding sequences which do not code for any proteins, but they will be very relevant to the, for example, some repetitive elements, sequences which are repeated in the entire genome several times. You can act as a biomarker. And also you can study the regulatory sequences which control the expression of genes. Similarly, in terms of protein, you can also identify the protein or can identify to which group or the family your unknown protein belongs to based on the similarity, or for the search for the structure of the protein. Similarly, the sequence searching is also important while you design PCR primers for PCR reaction. Now, depending upon the type of resources stored in a database, there are different types of databases available. They are primary sequence databases, which store DNA sequences, protein sequence databases that usually stores the protein sequence information, protein classification databases, then pathway databases, microarray databases, which store the image and the analysis data of microarray experiments. Microarray is a type of experiment which is done to study transcriptomics. Is to know the expression of genes, how many genes in a particular system are being expressed. Similarly, there can be separate databases or 2D-based databases that also stores the image information related to polyacrylamide electrophoresis. Similarly, we also have another kind of repository or biological databases which store the public literature. As you all know, if you download some papers, articles, you go to different public literature search engines like Google. A search engine which give you the resources or links to different websites which can give you the related references or the text or different kinds of resources for download. So, important public literature search databases include the Medline or PubMed and so on. Similarly, there is another kind of database that is called patent search database. As you know, for patenting, the first and foremost criteria is to have novelty in a particular method or a product. For novelty searching, you must have to search in the databases whether the kind of technology or the product or the method that you have developed has been already known. If it is already known, then you cannot claim for a patent. So that is why there are many resources created which are called patent search databases. Databases can also be classified into generalized and specialized. Generalized databases usually contain the basic biomolecules and their structures like DNA, proteins, carbohydrates, and three-dimensional structures of proteins. But on the other hand, specialized databases contain spatial sequences like ESTs, express sequence tag, STS, SNPs, RNA, genomes, protein families, pathways, microarray data, etc. Generalized databases, as I said, usually contain the basic biomolecules, their sequence and structure. Two main classes are there. For DNA, some examples are GenBank, which is hosted in NCBI at US, EMBL, European Molecular Biology Laboratory, or it is nowadays called ENA database, which is hosted in European Bioinformatics Institute in UK. Another important resource is DDBJ, DNA Data Bank of Japan. It is hosted in Japan in Asia. Then protein databases include some important examples are Probodh databases and PIR, protein identification resources. Interestingly, three most important databases, that is EMBL, or nowadays called ENA, GenBank, DDBJ, although they are hosted by three institutions of three different continents of the world, but they do have a very, very important integration among themselves so that resources are shared among the databases. And if you get access to one, you can easily download and have access to resources in the other. And these are the three most important resources for the biologist today. Spatial databases contain, as I said, information regarding certain spatial sequences like ESTs, STSs, SNPs, HTGS, and RNAs, etc. I'm not going to detail due to lack of time because we have a limitation of 40 minutes time. Protein family databases examples are ProCyte, PRINCE, BLOCK. Pathway databases include EMP, Pathway. Then there are many others. Then microarray databases like NX, Array Express, GEO, etc. Another classification of database is based on whether the sequences are uploaded to the database as it is, or it is purified and it is, so to say, examined for their accuracy. So if they are examined for their accuracy and many other facets like related literature and other annotations are added to the data, then the databases which host such kind of information, they are called curated databases. It can be primary or archival databases which store any information posted by any worker in the world in the unpure form, then it is called primary or archival databases where the data has not been curated or is tested and examined for accuracy and they have been annotated. Once they are curated, they may be placed in secondary databases. I have already told about curated databases. Now, let me give some examples of important databases. Like Probodh. Probodh is a curated protein sequence database, and it is having a high level of annotations and a minimum level of redundancy. What is redundancy? Redundancy stands for duplicacy. That means there are no duplicate data. It is a curated database, so all the information stored are already curated properly. They are examined and tested properly for accuracy, and the redundant and duplicate data are eliminated. It's a very good resource for protein sequence information. Similarly, DDBJ, DNA Data Bank of Japan, it contains DNA data, which is also having supported by the retrieval and analysis tools. Not only you can have access to data, but also you can download certain analytic tools for analyzing the data. Similarly, EMBL, it's a nucleotide sequence data. As I already said, it is hosted by European Bioinformatics Institute. It is a data contributor for individual genome projects conducted by different authors and the groups. This database releases all information in the updated form every quarterly. Similarly, another very important resource is called Ensemble, which is a comprehensive source of stable automatic annotation of human genome sequences and confirm gene predictions. Stack is another such kind of biological databases, which is a tool for detection and visualization of expressed transcript variations. So if you are working with functional genomics, then this is a very important resource for you. So this also gives you certain tools to study and analyze the sequences. TIGR gene indices, or commonly pronounced as Tiger gene indices, this is also a collection of transcribed sequences in a variety of organisms. Again, it is a very important resource for transcriptomics, or for that matter, the functional genomics study. Now, NCBI, as I already referred to, the full form is National Center for Biotechnology Information. So this is a center developed by National Institute of Health, Government of US, and under it, there are a number of resources available. And now, National Center for Biotechnology Information, or NCBI, is one of the most important resources for the biologist to get access not only to different kinds of biological databases that store DNA, RNA, and protein sequences, but at the same time, it also gives you access to public literature. It also gives access to different tools which can be used for studying basic and advanced bioinformatics. And all of us are highly dependent on the resources created by this particular organization. So they develop software tools for sequence analysis and also for many other facets of bioinformatics, including primer designing. Now, when somebody updates or uploads information to the NCBI, you have got access to it. It is anybody in the world can upload information in the form of sequence in a specific format to the NCBI resources, that is to GenBank. So there is a tool called Bankit. Another tool is called SequIn. These are the two tools which are given by NCBI to upload your data. So if you download these tools, you can easily format your sequence information and upload it in an accessible and acceptable form to the GenBank. So you see, now as I said, GenBank resources get doubled every 15 months. Now there are several millions and billions of nucleotide sequences, gene sequences, as well as complete genome sequences of different species in the GenBank. So when any information is put up by any worker in the world, that is first put up in the GenBank. So GenBank is like a, you can say, dustbin. Any type of information uploaded by any author in the world is first put into GenBank without curating it, without examining and testing it for accuracy and updating it. After it is accepted, you will be given an accession number, a specific unique ID by which you can claim that you were the original author of this particular sequence. It may be partial gene sequence, it may be a complete gene sequence, it may be a complete genome sequence. Once it is received by the GenBank, you will be given the authorship to it in the form of an accession number. Now after that, before being published, the sequence will be examined both by computational method, by using certain computational tools, as well as by a group of curators or examiners manually. By both the processes, the sequences will be examined and it will be given complete annotation, putting up information related to the sequence along with the sequence data. Then it will be referred to some other databases within NCBI, Say, for example, one such database is called RefSeq database. This RefSeq stands for referred sequences. This contains sequences which have been referred from GenBanks after annotation. Mostly it contains the messenger RNA sequences which have been curated both by manual as well as computational process. Now the question is, if you get access to the information stored in a GenBank, in what format it will be available to you? I think yesterday, those who have attended, they have already known what is the kind of format that is used for nucleotide sequences and the format that is used for the amino acid sequences. Now, as we know, DNA sequences contain 4 nucleotides, A, T, Z, and C. And according to the type of nucleotides in an array, in a particular segment of a DNA, that we refer to as functional part of the DNA gene, the final product will vary. I'll not go into detail about how it happens because yesterday it was discussed by Dr. Chetia in detail. Now, the sequence information of, suppose, a DNA sequence, how it looks like within a GenBank. If you download it from GenBank, it will appear like this. There are a lot of information pertaining to the sequence. Not only the sequence information as such, it also gives you different data pertaining to the sequence. Say, for example, the first part is called header. The header contains the specific unique name or the accession number of the sequence. Say, for example, this particular sequence is named as AY182241, and the total length of the sequence is 1931 BP. This is a messenger RNA sequence. Then it will tell you the source of this sequence, means the species from which it was obtained. It is a Melas domestica. Then a lot of other information will also be carried by the sequence data regarding the taxonomic status of the particular organism and the authors who have uploaded it to the GenBank. Then the title of the paper in which this sequence was included in the publication. And also there are some other important information. Say, for example, what type of sequence it is, and then it will also tell you the structure of the sequence, means whether it is a gene sequence. If it is a gene sequence, what is the part of the sequence which is really the coding sequence, means which code for the amino acids of the final product protein. Say, for example, for this particular gene sequence, although the total sequence length is 1931 BP, that means it contains 1931 nucleotides, but the actual coding sequence is starting from nucleotide number 54 and it extends up to 1784. And this is a gene which code for a protein which is named as terpene synthase. And if it is translated, then the amino acid sequence, corresponding amino acid sequence of this gene sequence will be like this. So this translation is done by computational process. There are bioinformatics softwares available. If you have got the gene sequence data, you can put up the information to the online and offline bioinformatics tools to get the translated product of protein. That means it can predict what kind of amino acid sequences the protein product of a particular gene have. And finally, it also provides you the entire sequence information. Okay. As I say, that EMBL, GenBank, and DDBJ are the 3 most important resources for the biology. you can download your required type of information from these databases for your job. It contains genome the product of the genome projects, that means the sequences obtained from study of the genome sequence of different organisms. It also has the information pertaining to many other biomolecules, including the protein sequences, the gene sequences, transcript sequences, and then specialized sequences like ESTs, STS, and many others. One interesting fact is that already more than 2 lakhs different species have been covered by these databases, and the sequences obtained from more than 2 lakh different species have been already uploaded to the online databases. And more than 1,000 new species are added every month. So you can very well guess that how it's going in a logarithmic way. Now the question is, when and how we will go to the databases? Suppose you have got a gene and you have cloned it and the sequence is available with you. Now you want to know, is it already in the databases? Then you can use certain tools. Say, for example, you can go for using a tool called Basic Local Alignment Search Tool, which is available in NCBI, to know whether your sequence matches to some other sequences in the databases already known. Then if you know, find that they are 100% identical to some known sequences already, that means your sequence is already known, and you can find out which particular species your sequence belongs and what characteristic it varies. If it is not 100% identical, you can search similar sequences in databases, and you can go for identifying percentage of identity and to which species your sequence may belong to. If it is not matching very highly to the sequences already in databases, then you can search for some parts of the sequence matching to some other sequences in database. That means if some regions of the sequence are conserved and they are highly similar to some other sequence, if not totally, then you can also get some idea what kind of information your sequence may carry. And by downloading these sequences with having a very high similarity and low similarity, you can also go for analyzing the data, compare them, and go for development of a phylogenetic analysis, drawing a phylogenetic tree, so that you can also establish the relationship between the species. That is called molecular phylogenetics. We shall take on this particular topic in a later class. First of all, in next class, I'll take on the sequence similarity search, or what we call sequence alignment, how the sequences are aligned. Then in the next, we shall discuss about the molecular phylogeny. Then similarly, we can also have information about protein sequences, protein structures. We can compare them. We can get the homologous protein sequences, download them, and can carry on many other computational biology experiments to discover new compounds and new drugs. We will definitely come to all these aspects in later course in our online lectures. And Dr. Chetia will also be taking some of the classes, and we'll be alternately taking classes for you, for your benefit, and you can put up questions. Now, at this point of time, I stop my lecture for today, and I make it open for you to question. I think there is very few minutes left, only 2-3 minutes left. You can put up your questions immediately. For that, you have to switch on your microphone, and if you have got any query, you can put up in 2 minutes.
Hello, sir.
Yes.
Sir, you told about something.
Yeah.
Yes, sir. I'm Jyotirmoy Das. Something you told about annotations.
Yeah.
Sir, at present I'm from Silabathar and I'm studying in Assam University, Silchar.
Okay.
Sir, you told something about annotation.
Yeah.
But I did not get the point, annotation.
Yeah, annotation means a processing of the sequences to associate the sequences with related information. Say, for example, when you draw a diagram, you normally go for labeling the different parts or components of the diagram, and you can describe those parts with a similar way. A sequence can be annotated by putting up information pertaining to different components of the sequence. Say, for example, the gene sequence. Which part is the regulatory sequence? Which part is the transcription search site? And what is the coding sequence? And what are the coded proteins? All these information will be tagged to the sequence information along with the published literature. And the entire process is called annotation.
Hello. Hello.
Sir.
Yes, I'm listening to you.
Hello. I'm Nipukumar Das.
Yeah.
So you told about generalized and specialized databases. Why RNA is not included in these generalized databases? Why it is included in specialized cases?
So normally, except for viruses, for other organisms, RNA cannot serve as a genetic biomolecule. Okay, it is genetic apparatus is in the form of DNA, and all database formation started with DNA. So DNA databases are called as primary databases or generalized databases, and RNA sequences are transfer RNA and ribosomal RNA are the product of DNA. Of course, we have almost reached 40 minutes time, so that is why RNA sequences are not included in the generalized databases.
Okay, sir. Thank you, sir. Thank you.
So, thank you.